This experiment will require these tools for proper procedure. Pipette sterile PBS into a microcentrifuge tube. This will be used to wash the colonies that are scraped off the plate. Check that the plate you are taking the sample from only contains one colony. It must be a pure culture. This first plate shows multiple morphologies, and thus it is contaminated and not pure. This plate only contains a single colony morphology and can be used for this extraction. Flame the loop to prevent contamination of the sample. Then scrape the colonies off the plate using the sterile loop. Place them into the microcentrifuge tube. Be sure to sterilize the loop again when you are finished. Next, vortex for 10 to 15 seconds in short increments. Keep vortexing until you no longer see clumps of cells. Always be careful when opening culture tubes after vortexing, as aerosols can cause contamination. Wash the cells by centrifugation of the suspension. Cells will go to the bottom. Remember that you must always balance the centrifuge by placing a tube containing the same volume of liquid in the opposite slot. Always put the cap back on the rotor, then set the centrifuge to 10,000 times gravity for 1 minute. Remove the supernat, which consists of the liquid above the cells at the bottom, leaving only the pellets. The pellet contains the DNA inside the cells. Discard of supernat in the proper biohazard bag. Pipette fresh sterile PBS to the sample suspending the pellets once again. Then vortex in short bursts until you no longer see clumps of cells. Rather than vortexing alone, it is sometimes easier to resuspend this pellet by pipetting up and down continuously. After resuspending the pellet, place the tube into a hot water bath set at 50 centigrade for 5 minutes. After heating in the water bath, glass beads will be used to increase shearing forces and to help lyse the cells. First, pipette all the sample into a microcentrifuge tube containing chem glass beads. Then vortex for at least 5 minutes. Now centrifuge again for 1 minute at 10 times gravity in order to pellet any unlysed cells and cellular debris. The DNA is now the supernat because the cells were broken open. Pipette the supernat with the DNA again into a collection tube and keep the tube. The next step is to use a genomic DNA cleanup kit. This kit will help remove proteins and other cellular components that may interfere with the PCR. Follow your TA's instructions according to the particular kit that you are using. To quantify the proper amount of DNA needed for PCR, DNA concentration is determined with a spectrophotometer. Clean the plate with a Kim wipe to prevent cross-contamination. Then carefully pipette the controls, water or buffer, onto one of the spots on the microvolume plate. Make sure to write down which slot you placed your sample. The TA will set up the software to read your sample by selecting the appropriate spots to read. After the absorbance of the samples is read, the computer will calculate the OD260-280 ratio, and the data can be exported to Excel. When you're looking at the spreadsheet, find the position of the sample you loaded and determine the DNA concentration of your sample. Calculate how much DNA you need for the PCR reaction. For the PCR mix,